Well, dear students, today we are going to discuss about the enzymes. These are the biochemicals which carry out the biochemical reactions. How they they speed up the biochemical reactions. May I tell you, people, that there is difference between the chemical reactions and biochemical reactions. The biochemical reactions are those reactions which happen to occur in the biochemical systems. While the biochemical reactions are those reactions. The chemical reactions are those which occur uh, in the non-biological systems. For example, if you if you increase the substrate or increase the enzyme, increase the substrate, uh, the 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 chemical reactions, the speed of chemical reaction would increase. Here, in case of the biochemical systems, if you increase the amount of substrate, it will the speed of reaction would increase. But after a certain level, there would be a constant speed. No increase would happen. Okay. Second, if you increase, for example, the temperature in uh, in a non-living system, in a chemical reaction, what will happen? The speed may increase, the rate of reaction may increase. But here, in case of enzymes, all the enzymes except for one example, ribozyme, all the enzymes are protein molecules. These are the biological active protein molecules. Okay, and the protein molecules, they are all protein molecules. The proteins, they are temperature sensitive, and these are globular proteins. So the globular proteins, they have a certain conformation. If that conformation is disturbed, what happens? The enzymes cannot act. Okay, they cannot regulate the biochemical reactions. So here is the difference between the biochemical reactions and chemical reactions. So you must note it. Secondly is uh, what is the name of that molecule which is acted upon by the enzymes? That is what the substrate. The substrate. Uh, any molecule or a substance which is any molecule or a substance which is acted upon by an enzyme, that is known as substrate. Okay, substrate can be one or more than one. Well, you can show this something with expression. How? Enzyme and substrate, that can be one or more than one. And what happens? Enzyme and substrate, they react, and enzyme substrate complex is formed. Okay, enzyme substrate complex. And then what happens? The product is formed. After the formation of product, the enzyme is separated. Enzyme is separated. Should I mention here to make you understand? And a verse of an Indian poet. He says, "What मैं तो गजल सुना के अकेला खड़ा रहा, मैं तो गजल सुना के अकेला खड़ा रहा, सब अपने अपने चाहने वालों में खो गए." What? The enzyme uh, makes the substrate to combine together, or it separates. If there is, for example, there is maltose, that is one substrate that is acted upon by an enzyme, that is maltase, and two products are formed. What? Two glucose molecules, they are separated. Okay, and sometimes the glucose molecules, they combine together, and one product is formed, that is what? Maltose. Okay, so this is how the enzymes, they break the biochemicals, or they unite the biochemicals together. Well, There are two types of the enzymes you must make sure. The enzymes, they are of two types. But the enzymes which consist solely of proteins. The others are there which have two parts. Those are what, the protein part and non-protein part. The non-protein part, that is known as what, the cofactor. The non-protein part of an enzyme is known as cofactor. And the cofactor, again, are of two types. What, molecular cofactor and metallic ions. Molecular cofactor and non-molecular, you can say. The non-molecular is what, that is metallic ion. That can be what, the magnesium ion, or that may be calcium ion and manganese, etc., etc. Okay, and these are known as activator. Now, second type of the non-protein part is what, the molecular part, and that is what, either prosthetic group or the coenzyme. If the non-protein part of an enzyme is covalently bonded with the protein part, then it is known as what, the prosthetic group. And for example, heme, okay, and heme, that is the part and parcel of hemoglobin, okay, the protein part, that acts as the enzyme, okay. And here is what, another molecular type of the molecular molecular type of the cofactor, that is what, coenzyme. The coenzyme, there are many coenzymes, okay. And the good example comes from what, nicotinamide adenine dinucleotide, NAD, or FAD, flavin adenine dinucleotide. These are the most important coenzymes, okay. Well, if sorry, if the the non-protein part is not covalently bonded, it is loosely bonded. What does mean loosely bonded? Should I mention here? Nicotinamide adenine dinucleotide may combine with an enzyme, okay, and then it would leave after the biochemical reaction is completed. Same is true with the FAD. Same is true with the coenzyme A, so and so. And this is what the meaning and message of loosely bonded. What about the activator or the metallic ions? These are like what, should I mention? Should I exemplify here? The prosthetic groups are like the wives. Okay, if you get married, your wife lives with you throughout the life. Okay, this is what the prosthetic group. Okay, and the coenzymes, these are like the maids. Okay, uh, she comes to your home, she works, and then after 1 hour she may go to another home, and after third hour she will go to another home, so and so. So nicotinamide adenine dinucleotide, FAD, and coenzyme A, and so and so. All these are coenzyme Q, etc., etc. These are what, the the maids. Okay, these are not covalently bonded. Okay, and the next is what, the activator. You're going along, okay, and your vehicle that stops working. Okay, what happens? You ask any person to help you, and he or she helps you and goes away. You never meet again. That is what, the metallic ion. Okay, that is the activator. So here is a story of the a whole story of the enzymes, their structure, and their way of function. One thing may clear once again. As I told you people, the difference in the beginning of this lecture, I told you people that there is difference between the biological system, the biochemical reactions, and the chemical reactions. In biochemical reactions, make sure always enzyme substrate complex is formed first, and then the products are formed and enzyme is separated. Okay. In chemical reactions, always two reactants, they react and the product is formed, no more enzyme is required. So here is the difference between the enzymatic reactions or biological reactions or biochemical reactions and the chemical reactions. Thank you very much.